Okay, so what I'd like to do is to just review stuff that we have done, and I would like to start at the earliest point, starting with quiz 1, and then move forward from there. And we'll cover as much as possible in the time constraint. Okay, so I think we started our course by doing a broad overview of the biotech industry, right? So the quiz 1 is about the broad overview. Okay, and so the first question here is to name a biotech product that has a medical use and one that is used in food industry. So for example, medical use, you can hold on, let me get the pencil here. Okay, for medical use, you can have insulin, for example, is a biotech product for medical use. Well, there are so many other products. Epogen, EPO is a biotech product for medical use. Human growth hormone is another, etc. And for food industry, used in the food industry, we started our course with making cheese with the help of recombinant renin, right? So that's the enzyme that is used in the food industry. You can also use many other enzymes are used in the food industry. For example, you might use some enzymes to clarify fruit juices. Okay, so enzymes to clarify fruit juices is another example. So many of the enzymes, okay, are used in food industry. Okay, second question, name two differences between a biotech product and a traditional pharmaceutical product. Now remember that biotech products are mostly proteins, right? And traditional pharmaceutical products are small molecules. So one difference is the molecular weight. Because proteins are relatively large molecules. So biotech products are large molecular weight, and traditional pharmaceuticals are small molecular weight. Okay, and what would be another difference between a biotech product and traditional pharma? Well, how they are made. For example, biotech products are made by biological agents like cells. Okay, so made by biological agents. They need not be cells, but usually some biological origin, right? Biological agents. Hope you can read my handwriting. Okay, and traditional pharmaceuticals are made by chemical synthesis. Okay, so you remember these differences. Biotech products are proteins, large molecules. They're made by biological agents like cells or mammalian cells or bacteria. Small molecules are made by traditional pharmaceuticals are small molecules made by chemical synthesis. Okay, and other differences, biotech products are usually injected, and they need a lot more cleaner atmosphere. You need to avoid contamination. Okay, so those are some of the differences between biotech products and traditional pharmaceuticals. Okay, the next question is place the following manufacturing processes in chronological order. So what comes first? Of course, first thing is fermentation. So this is number 1. Okay. Then after that is purification, which is number 2. And after purification would be formulation, because you need to formulate the product into the right buffer. And then you would fill the product, and then you would package it. Okay, so that's the appropriate order: fermentation, purification, formulation, filling, and packaging. Okay. Kevin and Isaiah, let me know if you have any questions or if you don't understand something that I'm talking about. Okay. I'm going to try to get through as many questions as possible so you have a good review. Okay. Now, only 1 out of 5,000 to 10,000 products screened by a company makes it as an approved pharmaceutical. Is this true or false? I think it's false because it's even larger. Anybody remember the number? The number? How many makes it to the makes it to an approved product? Well, you can look it up in the lecture. It tells you how many. Okay. And then what's the purpose of? Yeah, let me check. Okay, let me. I'll have to check. You should check the. I believe it's false. I think it's more like 1 in 100,000 or something like that. Okay, but verify from the lecture notes. Okay. Now, what's the purpose of phase 1 clinical trials? Phase 1 clinical trials is, if you remember, the purpose is to test safety and dose ranging. Okay, this is not to test efficacy usually. Okay, so you need to first test safety in humans. So usually phase 1 trials are conducted with very few people, mostly healthy volunteers. Okay, that's what it tested. And what data need to be provided to the FDA before you begin phase 1 trials? You need to provide toxicology data, animal studies. Okay, toxicology data. And you also need to provide manufacturing process, how you are manufacturing it under GMP conditions. Okay, so those are the things that you need to provide to the FDA. Now, what about phase 2 and phase 3? Phase 2, you expand. You are looking for efficacy. Okay, phase 2, you are looking for efficacy, but in a smaller number. So you have now shown safety, but now you're expanding the trial to test for efficacy with maybe 100 to 300 patients. Okay, and if that passes, then you go on to phase 3. Phase 3, you need to you expand the trial even further, and you enroll thousands of patients. Thousands of patients. Okay, and to enter phase 3, you have to show to the FDA that your phase 2 trial went well, that it met all its goals, and that your manufacturing process is okay. Okay, so phase 3 trial, you depend on phase 2 data to test thousands of patients. So are we clear on what is phase 1, phase 2, phase 3? Phase 1 is for safety and dose ranging with healthy volunteers. To go there, you need to provide animal toxicology data and manufacturing process. Phase 2, test for efficacy in small number of patients, and you need to provide data from phase 1 trials. Okay, and show that it worked, it is safe. And then phase 3, you expand the population to thousands of patients, and you need to provide data, efficacy data from phase 2. Okay, so that is quiz 1. So I'm going to load quiz 2 now. But before I go there, before I go to quiz 1, I want to talk about something else. Okay. Let's see. Okay, so when we started our labs, do you remember the first experiment we did? We talked about making cheese, right? Okay, and so in the context of this cheese lab, we also talked about the scientific method. Scientific method. So the scientific method, if you remember, involves 6 steps. You make some observations. Make some observations. Second, you ask some questions about it, about the observations. And then third, you're going to yes, okay. So what is it, Isaiah? What's the answer? Isaiah found the answer. 1 in 5 to 10,000 products are approved. Okay, so I stand corrected. So it is, so that was true, right? 1 in 5 to 10,000 products. Okay, thanks for checking on it. Okay, all right. So in the scientific method, you start with observations. You ask questions about your observations, and third, you would propose a hypothesis. Okay, and then you would um, test. Then you would make a prediction based on your hypothesis. You would make a prediction based on your hypothesis. And fifth, you would do testing. You would do experimentation. And then finally, based on the experiment results, you would draw some conclusion. Okay, so this is the scientific method. And so let's take the cheese lab, for example. Okay, what were some of the observations? Our observations might have been that if you use an enzyme, um, like renin that you get from the calf's stomach, it can curdle milk, right? It can curdle milk. So those were that might be an observation. So then you could ask the question: can I take the gene for that enzyme, insert it in a bacterium, make a renin based on that, and use that renin, use the recombinant renin to make cheese? That would be your question. And then you would propose a hypothesis that your hypothesis might be the recombinant renin will work just the same as natural renin. Okay, recombinant renin will work just the same as natural renin. That's your hypothesis. So what would be a prediction? A prediction would be if I curdle 10 milliliters of milk with natural renin and recombinant renin, I will get the same amount of curds, right? I will get the same amount of curd. And then you would do the experiment, and you will measure the amount of cheese being made, amount of curd being made, and you will test to see if the amount of curd was the same. Okay, so so is this clear, the scientific method and how we go about? You make observations, ask questions, propose a hypothesis, make a prediction, and do an experiment and draw conclusions. Okay. Now, when you do experiments, you have certain variables. Okay. So you have usually, okay, you're going to have a hypothesis, and then you're going to have certain variables. So one kind of variable is called the independent variable. Now, independent variable is the variable that you are actually changing. You are changing or testing this variable. Okay, so in our case, what was the independent variable? It was the type of renin, whether it is recombinant or natural, right? Recombinant or natural. And in fact, we had a third type of renin, which was the fungal renin, right? So we had three different types of renin. So in our case, the independent variable was the type of renin that we use. So we were looking for the effect of the type of renin on something. So what were we measuring? What were we measuring? We were measuring the amount of curds, right? Amount of curds. And so this, what we actually measure, is called the dependent variable. Something that the independent variable may have an effect on, right? So this is the dependent variable. Okay, so you have the independent variable. That's the one that you are changing. You are purposely changing that, right? Sometimes we also call it the manipulated variable. So you are actually manipulating the independent variable. You are going to look for the effect of the independent variable on the dependent variable. So in our case, the independent variable was the type of renin. The dependent variable was the amount of curds or amount of cheese that we are getting, right? So those are that's independent and dependent. Now, some things you want to keep the same. You don't want to confuse the experiment. So you want to, for example, you may want to keep the temperature the same, right? We don't want to have the recombinant renin at one temperature and the natural resin at another temperature, because you know then you wouldn't know whether it was the effect of temperature or the type of renin. So we want to keep the temperature the same. We want to keep the what else do you want? The volume of renin, you may want to keep the same, right? Renin or milk. So these are all variables that you want to keep constant, right? So these, we want to control these variables. So that's why these variables are called controlled variables. Okay, so controlled variables in an experiment are the ones that we keep the same for each condition. Okay, so we are testing the effect of the independent variable, which is the type of renin. Okay, and we are testing its effect on the dependent variable, which is the amount of curds or cheese. And when we are testing that, we are keeping the controlled variables constant. So we are keeping the temperature the same, 37 degrees. We are keeping the volume of renin the same, which might be, 5 milliliters or 1 milliliter, and volume of milk the same, which might be 10 milliliters. So in each case, we are going to keep these the same, and that's why we call them the controlled variables. Okay. Now, when we are doing this test, we are also going to make sure that the effect that we are seeing is real. How do you do that? We want to make sure that there is a control group. So there is a control group and an experimental group. What do we mean by that? So experimental group are the different types of renins, right? Different. So this is natural renin. This is recombinant renin. And the third may be fungal. Okay. So these are different types of renins. But we also want to know that that you are not going to get cheese just by adding water. Okay. So by adding just water, you're making sure that you're not going to get any cheese, right? Because if you get cheese even with water, then your experiment is flawed. Then the experiment doesn't work. So in any experiment, you should have a control group and the experimental group. So the control group refers to the negative control, where you do not add any of the independent variables. You do not have any of the independent variable. So in this case, the independent variable is the type of resin. Okay. The dependent variable is the amount of cheese or curds that you're measuring. The controlled variables, remember it's different than control group, right? Controlled variables are the temperature and the volume, right? And then the control group is an experiment where you just have water instead of the renin. Okay, instead of the renin, you have water. You add water to the milk and make sure that there is no cheese. Okay, so I just want to make sure that everybody understands the scientific method, what is a hypothesis and how you formulate a hypothesis. So for example, one of the hypotheses that we could formulate in this case is the amount of cheese formed, the recombinant renin will work the same as the natural renin. That might be one hypothesis. Also, you could have a different hypothesis. You might have a hypothesis which says the recombinant renin will work better than the natural renin because it is purer. Okay, so you have to have some rationale for making the hypothesis. Okay, so I might hypothesize that recombinant renin will work the same as natural renin because it has the same amino acid sequence. Okay, that's a rationale. Or I might hypothesize that the recombinant renin might work better than the natural renin because it is purer. Or I might hypothesize that the natural renin will work better because it is folded in its natural condition. Okay, so you could form any hypothesis, but you should provide some kind of a rationale, some educated guess. So your hypothesis is an educated guess based on some rationale. Okay, so the scientific method, you have observations, you make observations, you ask questions, you formulate a hypothesis, then you make a prediction. So in this case, we made a prediction that the amount of cheese that we measure, the amount of curds that we measure, will be the same in all these three cases. That's the prediction that we made. Then you run the experiment, you get the data, and then you conclude. And suppose you do get the same amount of cheese, 3 milliliters of cheese, then you would say my hypothesis was correct. Or if you don't get it, then you change your you say my hypothesis was incorrect, and then you go back and make another hypothesis or make more observations. Okay, that's the scientific method. So there will be a question on scientific method in the exam. Okay. All right. So how are you guys doing? Did you understand all these concepts in the context of our cheese experiment? Let me know if you have questions. Okay. All right. Now I'm going to load the quiz number 2. Okay, and we'll go through quiz number 2. Okay, so quiz number 2 was about safety. Okay, so is this true? The worker is solely responsible for safety in the workplace? Yeah, it's false. Remember, we talked about this, the workplace safety is a combined responsibility. So it involves OSHA, which is the Occupational Safety and Health Organization agency, that's the government. Then the company is also responsible. And each party has certain responsibilities, right? OSHA, the company, the lab itself, and the individual. So it's a shared responsibility, okay? Not just one person's responsibility. The lab is responsible for giving you instructions on safe practices. The lab is responsible for giving you the MSDSS, the material safety data sheet. Also is responsible for providing the safe safety equipment and personal protective equipment, right? The company is responsible for making sure that a culture of safety exists in the company, and that they make sure that they comply with all the laws. And OSHA is the one that enforces all the laws, right? Safety laws. So it's a shared responsibility. Which of the following is not an example of a non-government organization concerned with safety? So non-government is the key here. So there are many non-government organizations like American Society for Testing of Materials, Underwriter Laboratories, they do electrical equipment. ASME, American Society for Mechanical Engineers, they do pressure vessels. But this is a government organization. OSHA is a government organization, right? So remember that these are non-government. ASTM, Underwriter, ASME, etc. are non-government. Okay. All right. And OSHA. What are other government organizations? EPA is a government organization. Environmental Protection Agency. Also, Nuclear Regulatory Commission is a government organization concerned with safety as well. Okay. And of course, FDA is another government organization. Okay. So these are non-government and these are government organizations. Okay. Now, in the laboratory, where can you find data related to safe handling of a chemical? Remember, we talked about material safety data sheet, and give one example of data found there. Well, you might find the LD50 in there. What is LD50? The lethal dose 50. That means the dose at which 50% of lab animals were killed, right? So that's LD50. You might also find safety precautions to be taken. Also, you might find data on hazards, flammability, poison, different things. So, material safety data sheet. Okay. All right. Next. Okay. Name any two of the four quadrants of the NFPA diamond. Do you remember the NFPA diamond? And I forget how they divide that up. Okay. Now, okay, so combustion data, health hazards. Okay, so go ahead, Isaiah. You know all this stuff. Okay, so flammability, right? Flammability is one. Sorry, I can't write nicely. Health hazards. What else is there? Then there are some special hazards. And what's the last one? Maybe somebody can look that up for me. It's in the safety lecture. Okay. Oh, corrosive, right, corrosive. So, thank you. Okay, what would you do if you get a chemical splashed in your eyes? So you're going to go to the eye wash station, right? Eye wash station, wash with copious amounts of water, and inform supervisor. Inform somebody at least. Inform supervisor. Okay. All right. Why is it important to document, sign, and date all of your work in the laboratory? To document. So yeah, so you prove your research. Yeah, that's one thing. You know, from a patent standpoint, the first to invent is the person who gets the patent, and you will need to have proof that you actually did that. Secondly, it's important from a regulatory standpoint, because remember GMPs and GLPs. GLPs, good manufacturing practices and good laboratory practices. They state that you must document everything that you're doing, so that if there is any problem, if there is any issue, you can go back to make sure to figure out what happened, troubleshooting, and also for other people to follow what you did. So there are many reasons why. Okay. All right. So that was quiz number 2. I'm going to now load quiz number 3. Okay. All right. So this is math stuff. So let's do these things quickly. Perform the operations indicated. Now, negative 0.4 cubed. Now remember that when you raise something to a power, you multiply the same number by itself that many times. So it's negative 0.4 times negative 0.4 times negative 0.4. Okay. Now, when you multiply a negative number by a negative number, it's positive. So when you multiply a negative number by itself 3 times, it's going to be negative, right? So that's going to be negative 0.4 squared, cubed. And I'm going to take my calculator here and do the math. So it's 0.064, negative 0.064 plus 9.6 squared. So it's 9.6 times 9.6. So that's 92.16. And so that turns out to be 91.52, right? No, am I right? Hold on. No, 92.0096. Okay. Okay. Um, Now this other thing here, which says c raised to minus 3 raised to minus 5, when you are raising a power to another power, you multiply those two numbers. You multiply these two numbers. So this is going to be c raised to negative 3 times negative 5. So that's going to be c raised to 15, because negative times negative is a positive, right? Okay. Next, how do you convert numbers to scientific notation? Remember that scientific notation, you have to have the quotient, which is a number between 1 and 10. Okay, and then an exponent. So you have a quotient, which is a number between 1 and 10, times 10 raised to some number. Okay, so to convert this number to scientific notation, I can move the decimal place 3 places. So it's 2.03. So that's a number between 1 and 10. And then I have to multiply that by 10 raised to what? Because this is a small number and I'm making this to be a big number, I want to make the 10 raised to power less. So it's 10 raised to minus 3. Okay, 10 raised to minus 3. And how about this one? I have to move the decimal place the other way. 1, 2, 3, 4, 5, 6, 7, and 8. So 8 places. So it's 3.54 times 10 raised to 8. And this time it's positive 8, because this number is smaller than this number, so that this number has to be bigger than 10, and the other way around. Am I making sense? Anybody has a question about this? Okay. Now, how do we do this? What is this coefficient here if this is 10 raised to 6 and this is 10 raised to 2? Okay, so you know that this number is 4 orders of magnitude bigger than this number. 4 orders, because this is 10 raised to 6, this is 10 raised to 2. So I want to make this number right here, 4 orders of magnitude smaller than this number, to make those two equal, right? So that means I want to move the decimal place to the left 4 places. So if I move it 4 places, it's going to be 1, 2, 3, 4. So it's going to be 0.00645. Okay, does that make sense? 0.00645. Now over here, look at this. This number is bigger than this number. And how many orders of magnitude is it bigger? It's 1, 2, 3, 4. 4 orders of magnitude bigger, right? So this exponent number has to be 4 orders of magnitude bigger than this number. Okay, so let's do 10 raised to minus 3. 1 order bigger will be 10 raised to minus 2. Another order bigger will be 10 raised to minus 1. Another order bigger will be 10 raised to 0. And one more would be 10 raised to 1. So this is really 10 raised to 1. Does that make sense? Okay. All right. So common logarithm of the following numbers. Okay. So what's the common log of 1 million? Well, the way you determine logs, you can of course do it on your calculator, but 1 million is 10 raised to 6, right? 10 raised to 6. So the logarithm is that number to which 10 must be raised to give this number, right? So the common log of 1 million is 6. Okay. And what about this one? This is 1, 2, 3, 4, 5, 6. So this is 1 times 10 to the minus 6. And so the log of this number is negative 6. Okay. And you can verify that from with a calculator. Okay. All right. So now the next one says use a scientific calculator to determine log of this number. I'll let you do that. Calculator, it's pretty easy, right? Now remember that log is it's the natural, not natural log, but log to the base 10, right? We are always talking about log to the base 10. So in your calculator, there is log and there is natural log. Stay away from this one. Stay away from this one, okay? All right. Now, how do you do anti-logs? How do you do anti-logs? Anti-logs is nothing but 10 raised to this number, 10 raised to 8.7. Okay. So anti-log of 8.7 is 10 y raised to x 8.7. It will be like 50118723.6. Okay, it's a large number. Okay. Moving to the next one. Use a scientific calculator to convert the following hydrogen ion values to a pH value. Now remember that the pH is the negative logarithm of the hydrogen ion concentration. Okay, so basically what you have to do is to take this hydrogen ion concentration, and it has to be molar, okay, molar, moles per liter. Okay. So you take 0.34, you change the sign to negative, and sorry. You take the log of 0.34, take the log of 0.34, which is minus 0.468. Okay. So it's negative minus of minus 0.468. So that's a positive 0.468. Okay. And how about this one, 0.0065 molar? So I'm doing that right now on my calculator, 0.0065, and log of that is negative 2.18, and so the negative of negative 2.18 will be 2.18, positive 2 point. That's the pH. Okay. All right. How are you guys doing? Are you following? I need to know that you're following. Or okay. All right. Use a scientific calculator to determine the hydrogen ion concentration of the following pH values. Okay. So now you know that the pH is 4.75. So what you want to do is first to make it a negative number. Okay. So take 4.75. And now you're going to take anti-log of this. Anybody remember what an anti-log is? How do you get anti-log? You just raise 10 to that number, right? Yeah, 10 to that number. So I have 10. Where's my y raised to x? Okay, 10 y raised to x minus. Can anybody do that calculation for me? Oh, I have a 10 raised to x here. Okay. All right. Sorry, guys. Okay. 4.75 negative and 10 raised to x. So that turns out to be 0.00 0, and one more 0, 17783. Okay, so so that's how many molar is the pH concentration. Okay, that's how many molar is the pH concentration. So you can say it is, you can do 1, 2, 3, 4, 5. So you can say 1.778 times 10 to the minus 5 molar. Okay, and you could do the same thing with the 10.5. So get negative 10.5 and raise 10 to that number, and you will get the hydrogen ion concentration. Okay. Now, how tall is a person who is 6'6" in meters? 6'6" in meters. So you can just do 6 feet times 12 is 72 inches, and you add the 6 other inches to that, so that is 78 inches. And then each 1 inch is 2.54 centimeters per inch. So 78 times 2.54 equals 198.12 centimeters. Okay, and so you convert that into meters, it would be 1.98 meters. Okay, all right. Moving forward, the density of glycerol is 1.26 grams per milliliter. So what is the volume of 30 grams of glycerol? So this is a very easy thing to do if you can figure out the units. Okay, so remember that, so you have 30 grams of glycerol. Just pay attention to the units, and if you divide that by 1.26 grams per milliliter, so when this milliliter is going to go in the numerator, grams cancels out, so 30 divided by 1.26. So that's going to be 23.8. And what will be the units? It would be milliliters. Stay consistent with the units, and you'll be okay, right? Okay. Next question. If a solution requires a concentration of 12 grams of sodium chloride in 1 liter total volume, solution requires concentration of 12 grams of sodium chloride in 1 liter total volume, how much sodium chloride is necessary for 250 milliliters? So this is a ratio, right? So you want 12 grams in 1,000 milliliters. Is the same as how many grams in 250 milliliters? Okay, so you just cross multiply 250 times 12 and divide by 1,000. 250 times 12 and divide by 1,000. So that's going to be 3 grams. Okay, get that? All right, so these are the math problems that we just went over. Feel free to let me know if you don't understand something that I'm doing, okay? All right, so I think that was quiz number 3, was it? So now I'm going to go to quiz number 4. Okay, so I have to load quiz number 4. Okay. All right. So, which of the following is not used to measure mass? Which of the? So, a triple beam balance is of course used to measure mass. An analytical balance is also used to measure mass. A serological pipette is used to measure volumes, right? Serological pipette is used to measure volume. So that's the answer. Electronic balance is also used to measure mass. So it's serological pipette. Okay. All right. Convert the following volume to liters and microliters. Okay. Remember my B to S BS rule? Okay. When you go from bigger to smaller, you want to move the decimal place to the right. Okay. So if I want to convert this milliliters to microliters, then I want to move the decimal to the right, right? And how many places am I going to move? Milli to micro. Milli is 1,000 and micro is 1 millionth. So it's a difference of 3 orders of magnitude. So I want to convert, I want to move the decimal place 3 places. Okay. So if I move the decimal, so I'm going to move to the right. Move to the right. So here is the decimal place 1, 2, 3. So it's going to be 250,000 microliters. Okay, when you go from milliliters to microliters. Okay, now how about liters? Liters, you're going from milliliter is smaller than liter, so you're going from smaller to bigger. So you want to move the decimal place to the left, right? So here I'm going to move the decimal place 1, 2, 3 places. So that's going to be 0.25 liters. Okay, so 250 milliliters is 250,000 microliters and 0.25 liters. Okay. Now, describe how you take liquid into a micropipette and how you dispense it. Do you remember the 2 stops? So, first stop, you go to the first stop and take the liquid in. Okay, and to dispense, go to the second stop. Right? Because the first stop takes in the exact amount of liquid that has been dialed in, right? And the second stop, you push out an extra volume so that you push out everything that has been taken in. Okay, so first stop and second stop, remember that. All right, when measuring volume of a liquid in a graduated cylinder, so here is the graduated cylinder, and here are the markings. And here is your liquid. So what do? You look at the bottom of the meniscus. Bottom of the meniscus. Okay. So basically, here is the meniscus, and it has the bottom. Oops. Yeah. So you look at right there. That's where you measure the volume. Okay. Bottom of the meniscus. Okay. Okay. What instrument would you use to measure and dispense the following volumes? Vince, are you listening? All right. Which instrument would you micropipette would you use for this one? What instrument would you use for this one? That's correct. P1000. because P1000 will measure between 100 and 1000, between 100 and 1000. Okay. Now, how about 3.4 milliliters? 3.4 milliliters. But for this one, you could use one of two things. You could use a 5 milliliter serological pipette, or you could use a 10 milliliter graduated cylinder. Okay, graduated cylinder. Okay. How about 5.3 microliters? What would you use for this one? You could use a P20 or a P10. Okay. How about 2.5 liters? This is definitely a graduated cylinder, right? Okay. What number should be dialed into a P100 display, P100 display, if a volume of 350 microliters is to be dispensed? Any idea? So the tendency is to say 350, but you know that P100, the maximum you can measure with this is 100 microliters, right? And so you should not be using P100 for 350 microliters. Okay. Now, if you were using a P1000, what would you use for this one? So P1000, remember that 100 is the maximum, right? That's 1000. So to get 350 microliters, you would be dialing in 035. 035 is what you would be dialing in a P1000. Everybody get that? Because the maximum 1000 is 100. This is equivalent to a 1000 microliters. So to get 350 microliters, it's going to be 035. Okay. All right. Oh, it was a typo, P100. Okay. But if it were not a typo, then you would say this is the wrong pipette to use. You should use P1000. Okay. All right. When do you use an analytical balance, and when do you use a tabletop electronic balance? So remember, analytical has to be really small amounts. Tabletop for larger amounts. So rule of thumb, greater than 1 gram, use tabletop, and less than 1 gram, use analytical. Okay? Greater than 1 gram, tabletop, less than 1 gram, analytical. Okay. All right. What instrument would you use to measure the following amounts? Choose the amount that is likely to give you the least error for each measurement. So 350 grams of salt. 350 grams of salt. We just talked about this, right? So it's going to be the electronic balance, tabletop electronic balance. And this is a small amount, 35.69. So that's going to be the analytical balance. Okay? All right. Any questions on this quiz here? Okay. And I'm going to load quiz 5, and that's the last one I'm going to do because we're already out of time. But no, we are not out of time. Okay, I'm going to do the next quiz, and we'll see if we can do some more. Okay. All right. So All right, this is on spectrophotometry, right? A molecule of interest has lambda max of 530 nanometers. What kind of spectrophotometer would you use and why? Okay, so do you remember the range for, so here we were talking about UV spectrophotometer or visible, UV or visible, right? So this 530 is in the visible range. Visible range is anywhere between 300 to, I forget, 600 plus, okay, nanometers is visible. Okay, so that's why you're going to be using visible for this one. How about if your lambda max was 280? 280, then you're going to be using UV spectrophotometer. UV spectrophotometer. Okay, less than 300, you would use UV spectrophotometer. Okay. Describe the role of the blank solution in spectrophotometry. What solution would you use for a blank when you're measuring the optical density of a bacterial suspension? Okay, so the blank solution is to set the zero in absorbance, right? Zero for absorbance. So sets the background to zero in measuring the optical density, or in measuring the optical density, right? So so it should be an appropriate blank. Okay, so if you have a bacterial suspension, well, what is bacterial suspension kept in? It's kept in the media, right? So you should use the media that was used to grow the bacteria in, right? Media used to grow bacteria, not water, right? In this case, you will not be using water because the media itself has other molecules that might interfere with your spectrophotometry. So you want to cancel out the effect of the media. So that's why you would use the media for a blank. Also, if you are making a protein solution in a phosphate, in some kind of a buffer, then you should use the buffer as the blank. Okay. For protein solutions, use buffer as blank. So only when you are making a solution in water, then you would use water as the blank. Okay. All right. Next question. A UV spectrophotometer uses the following kind of lamp. What kind of lamp? Anybody remember? So there are two different types of lamps that are used. Tungsten is used for visible light. Tungsten filament lamp is used for visible light. And a deuterium lamp is used for UV spectrophotometer. A deuterium lamp is used for UV spectrophotometer. Tungsten is used for visible. Okay. Next. All right. Which part of the visible spectrum will have the lowest absorbance in a blue-colored solution, and why? Lowest absorbance. Remember that when something appears blue, it means that it is not absorbing that part of the spectrum, that part of the light spectrum, right? That's why it looks blue. It absorbs everything else except blue. So the blue part will have the lowest absorbance, right? And because it is not absorbing, right? It's not absorbing any blue color light. Okay. All right. Turbidimetry measures the light passing through a solution that is not Turbidimetry measures the light passing through a solution that is not scattered, filtered, transmitted, or all of the above. Um, yeah, so if it is scattered, it's not going to be measuring that. If it is filtered, it's not going to be measuring that. So if it is transmitted, that's what it's going to be measuring, right? Ah, so Kevin is saying that these are not the questions that we had on your quiz. Well, there are two classes, right? Monday and Wednesday classes. So some quizzes were given to the Monday. I've taken some quizzes from the Monday class and some quizzes from the Wednesday class. And I usually have slightly different questions for the 2-day classes. Does that make sense? But the topics are about the same. The topics are about the same. So for example, in your class, I might have said, you know, is another version of this. It might say something like, you know, light passing through a solution is scattered, filtered, transmitted, or all of the above. Okay? Okay. So what is a standard curve as it relates to spectrophotometric measurement of concentration of a molecule? Well, as you remember, the absorbance of a solution for a particular molecule is usually proportional to its concentration, right? So a standard curve is a plot of absorbance versus concentration of the molecule being studied. And usually you would get a linear response, hopefully. At some point it will level off, but this is called the standard curve, the plot of absorbance versus concentration. And you usually use a standard curve in its linear region, You don't want to do it over here. You don't want to measure it over here. You want to measure it in the linear region, okay? All righty. Any questions about this? Okay, so now what I'm going to have to do is either I have done quiz through quiz 5. I have not put quiz 6, 7, etc. on a PowerPoint. So, and this system allows me only to load PowerPoint. Okay, so if you want to wait a couple of minutes, I can quickly load quiz number 6 onto a PowerPoint and then import it. Is that okay with you? Or I can read you the questions. Okay, so it's going to take me a couple of minutes to load that PowerPoint. Okay, so hold on just a minute. Okay, we are here. Can you guys see the new quiz? Okay, so which of the following are mass volume concentration units, mass over volume? Okay, so let's see, microgram is mass, microgram is a unit of mass, and milliliter is a unit of mass, volume. So this is a mass volume concentration unit. How about milligram over gram? Well, both of them are mass, so it's not a mass over volume, right? And then grams over liter. Yeah, this is a mass volume concentration unit. Milliliters over gram, it's volume over mass, so that's not either. So these two are the correct answers. Okay. All right. Next question. So this quiz is on solutions, right? What mass of copper sulfate is needed to make 150 milliliters of this concentration, 100 micrograms per milliliter, right? So this is fairly simple. So you just keep track of the units, okay? 100 micrograms per milliliter times 150 milliliters. Now remember, the milliliters cancels out, so what you're going to get is micrograms, right? So it's 15,000 micrograms. 15,000 micrograms. Now you can tell me that, or you can convert into milligrams, which would be 15 milligrams. Got that? Okay. Next, give the decimal equivalent of the following percentages. Okay, so the decimal equivalent is quite easy. 10% would be, just move it 2 decimal places to go from percentage to decimal, right? So this would be 1, 2. So it will be 0.1 is the decimal equivalent. 1, 2. This would be 0.25. 1, 2. This would be 0.02. And this would be 0.005. Okay. All right. What? Oh, by the way, over here you have to describe how the solution is prepared. Now, do you remember how the solution is prepared? We are going to take 80% of the final volume. So in this case, instead of 150 milliliters, you're going to take 120 milliliters, dissolve 15 milligrams in 120 milliliters, and then you can bring to volume to 150 milliliters. Okay, remember that description, how to describe how to make the solution. Okay. All right. What mass of sugar is needed to make 200 milliliters of a 3.5% solution? Okay, so 3.5% solution. The way to do this, I taught you 2 different methods, right? One way is to convert this into a decimal, and then multiply by the volume, okay, to get the grams. So 3.5, what's the decimal equivalent of 3.5? It's going to be 0.035. That times the volume is how many grams you're going to get. Okay, so that's going to be 7 grams. 7 grams of sugar. Okay, the other way to figure that out is 3.5% is 3.5 grams in 100 milliliters. Okay, so it's 3.5 grams in 100 milliliters. And so how many in 200 milliliters? So it's going to be 200 times 3.5 divided by 100. Okay, so that's going to be, by the way, this is milliliters, this is grams. So it's going to be 7 grams, right? Okay. All right. Next. Okay. All right. What mass of salt is needed to make 250 milliliters of a 5% salt solution? What mass of salt is needed to make 250 milliliters of a 5% salt solution? So again, and describe how to make the solution, right? So it's going to be, convert this into into a decimal, 0.05 times 250. So that's going to be 12.5. What will be the units of that? 12.5 grams, right? 12.5 grams. So how do you make the solution? You're going to dissolve 12.5 grams in 80%, so about 200 milliliters, and then B to V, bring to volume, right? Okay. So remember, whenever you're making solutions, you initially dissolve it in 80% of the final volume. Once you have dissolved it, then you can bring to volume to the final volume. Okay. All right. What's the molecular weight of these compounds? It's, I will normally give you the atomic numbers, for atomic weights for all the different different atoms, the different elements. So H2SO4 is going to be, H is 1, so it's 1 plus 1, that's H2, plus sulfur, I forget, I think it's 30 something, 32, plus oxygen is 16, so that's going to be 64. So 64, 96, so it's 98, right? Okay. And then calcium chloride is going to be calcium, which is what, 40? Calcium is 40 maybe, and chlorine is 35, so 35 times 2 is 70, so it's about 110. Okay. Very good. Somebody mentioned that it should be grams per mole. Thank you for pointing that out. Grams per mole. Okay. All right. What mass of sulfuric acid is needed for 0.5 liters of 0.5 molar sulfuric acid solution? So these are molar solutions, right? So now, let's say that I've given you the molecular weight of sulfuric acid, which is 98, right? So it's 98 grams per mole times 0.5. Now, capital M stands for moles per liter. So it's moles per liter times 0.5 liters. So liters cancels out, moles cancels out, so you're left with grams, right? So 98 times 0.5 times 0.5, so that's 24.5 grams. Okay. All right. What mass of calcium chloride is needed to make 700 milliliters of 120 millimolar calcium chloride? So now the twist here is, I'm using these milliliters and millimoles. So you have to make sure that you either convert them to moles and liters, or you take into account the milli. Okay. So let's try that. So 120 millimole is the same as 0.12 moles, right? 120 millimoles is the same as 0.12 moles. So 0.12 moles per liter calcium chloride times 700 milliliters is 0.7 liters. 0.7 liters. And we need the molecular weight of calcium chloride, which is 110 grams per mole. So liters cancels out, moles cancels out, and so you get 110 times 0.7 times 0.12, which is equal to 9.24 grams. Okay. How are you doing? Is that jog your memory? Okay. All right. So that's where I am at quiz 6. Do you want me to go on to quiz 7? Do you have time? So I'm going to have to load that. Okay. Hold on a minute, okay? Okay. Can you see it's quiz number 7 now? Okay, so somebody said, can I ask a few questions from other later quizzes? Of course you can. All right, so let's do this quiz and then you can ask your questions, okay? All right, describe how to prepare 100 milliliters of a 2 milligram per milliliter protein solution from a 10 milligram per milliliter stock solution. Now remember, you can always use this whenever you see stock solution. It means it's a dilution problem. You're doing dilution, right? And what's the big equation for dilution? C1V1 equals C2V2. And all you have to do is to figure out 3 of these things and find the 4th. Okay? So C1V1 is the original solution, the stock solution, right? So what is C1? 10 milligram per milliliter. That's the C1, right? And we don't know V1. We want to find out V1. And C2 is 2 milligrams per milliliter. This is C2. And V2 is 100 milliliters. Right? So then V1 is going to be C2 times V2, which is 100, divided by C1, which is 10. So that cancels, and you get 20. What would be the units for this? It's the volume, right? Milliliters, of course. Yeah, milliliters. So you need 20 milliliters of the stock solution to make 100 milliliters of the 2 milligram per milliliter, right? So you're going to take 20 milliliters and then bring the volume up to 100 with the diluent. Okay. What's the difference between independent and serial dilutions? Okay. So independent is, let's say that you have a stock solution of 100 milligrams per milliliter. Okay. And you make a dilution of it to 2 milligrams per milliliter. And you take the same stock solution and you make another dilution to 50 milligram per milliliter. So you're starting with the same stock solution. You make two different dilutions. Those are independent dilutions because each time you're starting with the same stock solution. Okay. Now serial dilution would be if you took this solution and made it into diluted it to 50 milligrams per milliliter. And then you took this diluted solution and used that to make a dilution of 2 milligrams per milliliter. Okay, so you're doing a serial dilution. You're going from here to here, right? Serial dilutions. Those are dependent dilutions. Now, serial dilutions, usually you have the same dilution factor. So in this case, we didn't have the same dilution factor. But for example, I can give you an example of how will you make a 1 in 1,000 dilution in one step and 1 in three-step serial dilution. Okay, so let's say that you have 1,000 milligram per milliliter stock solution, and you can take 1 milliliter of this and add 999 milliliters of diluent, and that's going to be a 1 in 1,000 dilution, and this is going to be a one step, right? One step. On the other hand, you can take 1,000 milligrams per milliliter stock and dilute it 1 in 10. Okay, so you could take 10 milliliters of this and add you take 1 milliliter of this and add 9 milliliters of diluent. So that's going to be a 1 in 10 dilution, right? And then you take 1 milliliter of that and add 9 milliliters of water. And that's going to be a second 1 in 10 dilution. And you do the same thing again, 1 milliliter of that and 9 milliliters of water. So that's going to be a third 1 in 10 dilution. And so the overall dilution, this final dilution is going to be 1/10 times 1/10 times 1/10, which is a 1 in 1,000 dilution. So this is called a serial dilution. So you had three 1 in 10 serial dilutions that accounts for 1 in 1,000, and here is a one-step 1 in 1,000 dilution. Okay. All right. How do you prepare 5 liters of 1x strength TA buffer from 50x stock solution? Remember, same thing. C1V1 equals C2V2. Whenever you see the word stock solution, it's a dilution problem, right? So what is C1? It's 50x, right? The stock solution. We don't know what is V1. We know V2 is 5 liters and C2 is 1x. So V1 is going to be 1x times 5 divided by 50. So it's going to be 0.1 liters. Okay. So how do you prepare it? You take 0.1 liters or 100 milliliters of the stock solution and add diluent to make it up to 5 liters. Okay. All right, let's go to the next slide. All right, describe how to prepare 1 milliliter solution of a 20 micromolar KCl solution from a 5 millimolar stock solution. Okay, so here you have to be just more careful of your units. Okay, see how I have millimolar here and a micromolar over here. So you should convert it either into a millimolar or micromolar. Be consistent. Okay, so and molar is moles per liter, and this is 1 milliliter. You understand? All right. So this is your C2. So your V1 is going to be, this is your C1, and I'm going to convert it into millimolar. 20 micromolar would be 20 micromoles per liter. So millimolar would be 0.02 millimoles per liter. Okay, and so it's moles per liter, right? Millimoles per liter. So you want to multiply that by 1 milliliter, but convert it into a liter. Okay, so it's going to be 0.001 liters, and then divide that by 5 millimoles per liter. Okay, and so you can do that calculation yourself, right? Okay, so that's how many liters you're going to get. And with the help of a diagram, show how you will make the following dilution, starting with a 125. So here is your stock solution, 125 milligrams. And so what you're going to do is serial dilution method. You're going to take 1 milliliter of this, and put it in a tube, adding 4 milliliters of diluent. And then, so this is your first 1 in 5 dilution. Okay, and so it's going to be this is going to be 25 milligrams per mil. And then you take 1 milliliter of this, into a tube, and add 4 milliliters, and you're going to get 5 milligrams per milliliter. And the same thing again, 1 milliliter and 4 milliliters. So this is the serial dilution concept, okay? All right. So I think we are running out of time. So somebody had questions about other related quizzes. Can you can ask me those questions now. Okay, so the question is pH of 1 molar acetic acid higher than 1 molar hydrochloric acid. Okay, why is the pH of 1 molar acetic acid higher than 1 molar hydrochloric acid? Okay, so you know that acetic acid is a weak acid. Is weak acid. And so it does not dissociate completely. Okay. In water. Whereas hydrochloric acid is a strong acid and dissociates completely. Okay. So when you get 1 molar HCl, it dissociates into 1 mole of hydrogen ion and 1 mole of chloride ion. Okay. So the molarity of hydrogen ion concentration in when you have hydrochloric acid is 1 molar. Okay. But acetic acid is a weak acid. So this CH3COOH, that's the formula for acetic acid. You don't need to know this. You just need to know that it's a weak acid. So when it dissociates, you only get the smaller amount of CH3COO minus and H plus. So you do not get 1 mole of hydrogen ion. You do not get 1 mole of hydrogen ion. Whereas with hydrochloric acid, you get the whole 1 mole of hydrogen ion because it completely dissociates. Okay, so you get hydrogen ion concentration that is smaller, that is less than 1 molar in case of acetic acid. And when you have lower hydrogen ion concentration, the pH is higher, right? Lower hydrogen ion concentration means pH is higher. That is why acetic acid pH, the pH of 1 molar acetic acid is higher than pH of 1 molar hydrochloric acid. It's because it is a weaker acid and it dissociates less than hydrochloric acid. Does that answer your question? Okay. Any other questions? Okay. So what are the 4 classes of macromolecules? 4 classes of macromolecules. So the first class is carbohydrates. These are mostly polysaccharides, right? Polysaccharides. That means they are polymers of sugars, and they are usually sources of energy. Okay. The second one is lipids. That means these are fats, fats and steroids and phospholipids. Okay. And they are also energy sources and provide insulation. Third class of macromolecules is proteins. And these are polymers of amino acids. And the fourth class of macromolecules is nucleic acids. Nucleic acids are DNA and RNA. They are nucleic acids, right? Deoxyribonucleic acid and ribonucleic acids, DNA and RNA. Okay? So those are the 4 classes of macromolecules. So nucleic acids have the information. Proteins is what is made from the information that is included in the nucleic acid, in the DNA, right? Okay, more questions. How are the polypeptide chains held together? How are the polypeptide chains held together? In what? Oh, in antibodies. Okay, in antibodies. So they are held together. Remember the polypeptide chain. In antibodies you have 2 heavy chains and 2 light chains, right? 2 heavy chains and 2 light chains. So these 2 are the heavy chains, and these 2 are the light chains, and they are held together by disulfide linkages, disulfide bonds. Get that? They are held together by disulfide bonds. And this region of the antibody is called the variable region. And this is the constant region here. Two heavy chains, 2 light chains. Okay. Any other questions? Okay. Question is, what distinguishes amino acids from one another? Okay. So amino acids, if you recall, are made up of an amino group and a carboxylic acid group. And it has an R group. So all amino acids have the amino group and the carboxylic acid group. But R group changes from one amino acid to another. So this is the R group is what distinguishes one amino acid from another. Some R groups are hydrophobic, some of them are hydrophilic, some of them are positively charged, some of them are negatively charged. Okay? In different structures. So this is what changes from one amino acid to another. And there are 20 amino acids, 20 different amino acids in nature. So you can infer that there are 20 different R groups, right? 20 different R groups. That's what changes. Okay, good questions. Any more? Okay. Anybody else? So those were all questions from Kevin. Any other questions? If not, then I am going to, see you tomorrow. If not, I'm going to stop the recording, and if you have questions, feel free to email to me, and I will try to respond before tomorrow's exam. Okay, but no guarantees right now. Okay, talk to you later.